You know, when a really dangerous fungal infection hits, the clock starts ticking, fast. The whole battle for a patient's life? It's often won or lost right there in the lab, in this high-stakes race to figure out exactly what the microscopic enemy is. So come on, let's follow the clues and dive into this incredible diagnostic journey.
All right, so here's our roadmap. We're going to follow the evidence through 5 key stages. First, we'll get a sense of the high stakes. Then, we'll see how the crucial clues are collected. After that, it's a first peek under the microscope. Then we grow the evidence in the lab. And finally, we'll crack its code for the final ID. So, first up, the hunt itself. And you got to understand, this isn't just some routine test. For a lot of patients, this is a full-on detective story, where every single minute, every tiny clue is absolutely vital. Which brings us to the big question, right? Why the mad rush? Well, fungal infections are a huge, and I mean huge, global health problem, especially for people whose immune systems are weakened. Getting the diagnosis right and getting it fast, well, that can literally be the difference between life and death.
And the answer, really, it all comes down to targeted therapy. See, not all fungi are the same, and the drugs we use to fight them aren't either. It's not one size fits all. Identifying the exact species tells the doctors which weapon in their arsenal is going to work. A quick, accurate ID means the right drug gets to the patient faster, which means better outcomes, and, this is a big one, it helps us stop the spread of really nasty, drug-resistant bugs.
Okay, on to the first step of any good investigation. You have to collect the evidence. Because honestly, everything that happens next in the lab completely depends on the quality of that first sample that comes through the door.
It's a simple rule, but in the world of mycology, that's the study of fungi, it is everything. A good sample is clean, it's taken from just the right spot. A bad one, though, one that's full of contamination, it can send the whole investigation down a completely wrong path, wasting precious time.
And this evidence, it can come from just about anywhere on the body. Depending on what kind of infection is suspected, labs get all sorts of things. Skin scrapings, hair, nail clippings, even deep lung mucus called sputum, blood, urine, or swabs from the mouth or other areas.
So let's take a classic example, skin scrapings. The process has to be super meticulous. First, you clean the area with alcohol to get rid of any bacteria just hanging out on the surface. Then, and this is key, you scrape the active leading edge of the rash. Why there? Because that's where the fungus is alive and growing. Finally, you pop those little scales into a sterile container and it's off to the lab.
So, the evidence has arrived at the lab. Now it's time for that first look. This is called direct microscopy, and it gives the team their first big clues. It can often give them a presumptive diagnosis in minutes, which is a huge head start while the more definitive tests get going.
Now, the absolute workhorse for this is something called a KOH mount. Potassium hydroxide, or KOH, is basically a chemical bulldozer. It just dissolves all the background noise. You know, the skin cells, the proteins, all that stuff. And what's left behind? Only the fungal parts. It works because fungal cell walls are made of this really tough stuff called chitin, which just shrugs off the chemical attack, letting it stand out so we can see it.
And the best part? The procedure is beautifully simple. You put a tiny bit of the specimen on a slide, add a drop of KOH, put a little cover slip on top. You can even warm it up a little to speed up the clearing process. In just a few moments, it's ready for the microscope. It's an incredibly fast and really powerful first step.
Now, it's really important to know the difference between our two main tools here. Think of it this way. The KOH mount is for clearing a sample that came directly from the patient. Its whole job is to answer one question. Is there any fungus here? Lactophenol cotton blue, on the other hand, is for staining a fungus we've already grown in the lab. It actually kills, preserves, and stains the fungus's structures so we can get a really detailed look at what it is. But if you want an even more sensitive look, this is where things get really cool. Labs can use a stain called chalcofluor white. It's a fluorescent dye that sticks specifically to that chitin in the fungal walls. And under a special microscope, the fungal parts literally glow this brilliant, almost ghostly blue-green. It's just amazing. And it's so sensitive, it can find the fungus even when there are only a few cells there. So, while the microscope gives us some awesome, fast clues, the absolute gold standard for a definitive diagnosis is culture. This is where we literally grow the evidence, creating a pure, living colony of the suspect that we can study in much, much more detail.
And the foundation of any mycology lab, and I mean this has been the case since 1910, is this stuff. Sabouraud dextrose agar, or SDA. This was designed specifically for fungus. The recipe, a ton of sugar and a low acidic pH, is basically a 5-star hotel for fungus, while being a really hostile place for most bacteria. It's genius.
But we can make it even more exclusive. Think of it like putting up velvet ropes at a nightclub. By adding an antibiotic like chloramphenicol, we tell most bacteria, nope, you're not on the list. Then, by adding a special antifungal, we block all the common, fast-growing environmental molds, the party crashers. This clears the dance floor for the real culprits we're looking for, letting them grow without any competition.
So as the fungus grows, and this can take days, sometimes even weeks, the microbiologists become detectives all over again. They're looking at the colony's unique features. What's the texture like? Is it cottony or maybe velvety? Is the surface flat or all wrinkly? And what about the color, not just on the top, but critically, what color is it on the reverse side? All of these are clues to the bad guy's identity.
Okay, let's move on to the final phase. And this is where technology has just created an absolute quantum leap. This is where we crack the fungal code, going from an ID that took days or even weeks down to one that takes just a few minutes.
You see, for decades, the old way of doing things was to run a whole bunch of biochemical tests. You'd see what sugars the fungus could eat, that sort of thing. But oh my gosh, these methods were incredibly slow, the results could be tricky to interpret, and frankly, they just didn't work very well for a lot of species. It was a slow, frustrating process.
And then everything changed. This technology called MALDI-TOF mass spectrometry came along. And as one scientific paper put it, its introduction was nothing less than a paradigm shift. It just completely, totally revolutionized the modern mycology lab.
So what is this thing? Well, to put it simply, instead of looking at what a fungus does, it looks at what it's made of. A laser zaps a tiny sample, which basically vaporizes the fungus's proteins. Then a machine measures how fast those proteins fly through a tube. This creates a completely unique protein fingerprint for that fungus, which is then instantly compared to a massive database for a positive match. It's like CSI for microbes. And the impact of this? It is staggering. I mean, just look at this. A process that used to take anywhere from 1 to 3 days can now be done in minutes from a single tiny colony. This isn't just a small improvement. It's a complete game changer in diagnostic speed. And this brings us right back to the patient, which is what this is all about. Before, a slow diagnosis meant doctors had to start treatment with their best guess, a broad-spectrum drug. But now, with a MALDI-TOF ID that comes back in minutes, they can start a rapid, targeted therapy with exactly the right drug, sometimes on day 1. It just enables quicker, life-saving decisions and can dramatically improve a patient's chance for survival.
So, this incredible journey, from a simple skin scraping all the way to a protein fingerprint, has totally transformed our ability to fight these diseases. And it leaves us with this really powerful question to think about. As this technology gets even faster and the databases get even bigger, what new fungal threats will we be able to spot and neutralize before they even get a chance to spread?